Now that we've talked about prokaryotes, we're going to continue on and talk about eukaryotic organisms, like these mating frogs that we see here. Before we get into that, though, I think it's important that we spend a little bit of time talking about sex, and also meiosis, which is the special type of cell division that makes sex possible. Sex was a major innovation in the eukaryotes, and it's very important in classifying the different groups of eukaryotes, as we'll see. So what is sex? Now that might sound like a really simple question. It's reproduction, of course. But it's not the only way to reproduce. Technically speaking, sex is where two individuals combine their genetic material to create a new individual. So for instance, you are the product of your parents combining information. Your father donated a sperm, your mother donated an egg. They each donated an entire set of information, and the zygote, the single cell that resulted and gave rise to you, had two sets of information. And all of the cells in your body have two sets of information. They have one set of chromosomes from your mother and one set from your father, and they're still separate in every single cell in your body. The exception, of course, are your gametes, your sperm or your egg. They only contain one set, and we'll talk about how that works. Not all eukaryotes reproduce using sex, and some alternate between sex and asexual, non-sexual reproduction. There are many eukaryotes that only engage in asexual reproduction, and the very first eukaryotes would have not been using sex, meiosis, to reproduce. So let's start by taking a look at asexual, non-sexual reproduction. And I'm going to give you a very simple hypothetical cell that only has two chromosomes. Now realize in humans, the sperm and the egg each contain 23 chromosomes, and your body cells, the cells in your liver and brain and so on, contain 46 chromosomes. I'm not going to try to draw all that out. So we have a hypothetical single-celled eukaryote here that just has two chromosomes. And notice that the chromosomes are different. They're different lengths. They contain different information. They contain different sets of genes. Okay, now what's going to happen just prior to asexual cell division, which is known as mitosis in eukaryotes, is that the DNA is going to be replicated. Before we get to that, let me just introduce a new term here, ploidy. Ploidy refers to the number of sets of chromosomes in a cell. If we have a haploid condition, that means that we only have one set of chromosomes, and we represent that as 1N, or simply N. Okay, so our haploid cell here is going to get ready for division, and before it does that, it has to replicate its DNA. So right now, those two chromosomes, they each contain one very, very long coiled up piece of DNA, but the DNA is going to be copied by DNA polymerase, and we end up with something like this. We still say that we have two chromosomes, but now each of the chromosomes is made up of two pieces of DNA instead of one. Next, those chromosomes are going to migrate to the center of the cell. The cell is going to elongate and split, and as it does, those chromosomes are going to be segregated. They're going to be packaged into the new daughter cells. So we start with a parent cell that is haploid. It has one set of information. The DNA is copied, then it's divided and split into two daughter cells, which are both, again, haploid. If everything goes as it should, the cells we end up with are identical to the cells we started with. All right, let's take a look at sexual reproduction. It's a bit more complicated. Here, we start with what's called a diploid cell. Diploid means there's two sets of chromosomes. So what I've tried to show you here are that we had one set of chromosomes coming from the mother, let's say those are the red ones, and we had one set of chromosomes coming from the father. But note that we have two long chromosomes, two short chromosomes. We have the same information from mother and father, although with slight differences, slight variations from gene to gene. But we have these two long chromosomes that we say are homologous. They contain the same information. We have two short chromosomes, and they're homologous. They contain the same information as well. This is called the diploid state, di meaning two, because we have two sets of chromosomes. Hap, by the way, means half. It's half of that. So we're going to start out with a diploid cell in this case. And again, all the cells in your body, apart from your gametes, your sperm and your egg, are diploid. The diploid cell is going to undergo DNA replication, and we end up with this. We started with 4 chromosomes. We still have 4 chromosomes, but now each chromosome consists of 2 strands of DNA instead of 1. This special cell division that's going to happen next is known as meiosis. And there's 2 steps to it. There's actually 2 divisions. Now, before we actually divide up the DNA, what's going to happen is the homologous chromosomes group together in something called a tetrad. So the 4 long chromosomes all come together, the 4 short chromosomes all come together, and there's swapping of information between them. And I've represented that by kind of blending the colors together here. In your body cells, in your skin cells, your liver cells, etc., you have 2 separate sets of chromosomes, one from your mother, one from your father, and they have not mixed. They do not mix until you produce your own gametes, until you produce sperm and egg, or egg, of course, in preparation for the next generation. Okay, so we have our cell here with our replicated DNA, and what's going to happen now is the chromosomes are going to be pulled apart. So we have these chromosomes that still consist of 2 parts. They're being pulled apart. And now there's going to be a second division. Again, this is all part of meiosis, without the DNA being replicated in between. And what we end up with down the bottom there are 4 haploid cells. So we double the number of cells, and then we double it again, and we cut the ploidy from 2 down to 1. Why does meiosis do this? Well, the whole point is that now we can take one of those gametes and we can combine it with a gamete, again a sperm or an egg, from another individual. Those 2 cells will fuse, merge together, and we go back to what we started with. So imagine that this gamete encounters another gamete from another individual, and they come together and they merge. And we now have a 2N cell, this is known as a zygote, that can begin the next generation. Just as a quick refresher, before we go on with the eukaryotic condition, remember what happens in prokaryotes. In bacteria and archaea, instead of having lots of separate chromosomes that are all straight, we have one massive loop of DNA. The chromosome is one big twisted loop of DNA in the center of the cell. That DNA is replicated by DNA polymerase, And then the two loops stick to opposite ends of the cell. The cell elongates, that pulls the loops apart. Very, very quick process. There's no nucleus to break down. There's no chromosomes to line up and segregate, etc. And this is why bacteria can reproduce so quickly in 20 minutes. Bacteria, if you remember, they can also do something called conjugation. And conjugation is sometimes erroneously called bacterial sex. Remember, there's a pilus, kind of like a tube, that they use to pass information from one cell to the next. And that's sometimes called a sex pilus, which is kind of unfortunate because, again, it's not sex. Sex is where you take two sets of information from two parents, as we just talked about, combine it and create a new individual. And this is not what's happening. We're just swapping little bits of information between individuals. I think I used the example before that if we were capable of this, you could maybe reach out your arm and touch the person next to you with your finger and transfer them just a little bit of information, maybe change the color of their eyes. Right? Of course, we can't do that. But that's conjugation. Conjugation is known as horizontal gene transmission because you're transmitting information to other cells of the same generation. Sex, and also any type of cell division, is a form of vertical gene transmission. Vertical meaning that you're passing it from generation 1 to generation 2 to generation 3. You're passing information through time. So in bacteria and archaea, we have asexual reproduction that gives us our vertical gene transmission, and that's referred to as binary fission in the prokaryotes. And then we have conjugation, which is just this weird thing where they pass information back and forth between members of the same generation. But they don't have mitosis, they don't have meiosis, and they don't have sex. Those are things that are confined to the eukaryotes. Let's take a quick look at the stages of mitosis in eukaryotic cells. Most living eukaryotes, not all, but most, follow the set of stages that we can see on the right. We have interphase. Interphase is the phase between divisions. So most of the cells in your body that aren't currently dividing will hang out in this phase until they get a signal from your body that they should divide. If they get that signal, then they'll enter into something called S phase, synthesis phase. It's part of interphase where the DNA is replicated. And after that, their chromosomes will consist of 2 pieces of DNA instead of 1. Then they enter into prophase, and prophase is where the nucleus starts to break down. The DNA condenses, it gets shorter and more compact. It actually becomes visible under the light microscope. In metaphase, the chromosomes move to the middle of the cell and they all line up in the middle of the cell. As all of this is happening, something called the mitotic spindle is formed, and it consists of microtubules that radiate out from each of the poles of the cell, so from opposite ends of the cell. The chromosomes attach to the spindle, and the chromatids, the individual parts that make up each of the chromosomes, are pulled apart, and they move to either end of the cell. Then during telophase, so anaphase is where the chromosomes are pulled apart, telophase is where they arrive at the ends of the cell and the nuclei are reformed. And then the last step here, and it overlaps with telophase, is the cell elongating and pinching off into 2 separate cells. Let's quickly take a look at the evolution of this whole process. It is a very complex process. There's lots that has to happen. It's slow. It takes a lot longer than binary fission in bacteria cells. In dinoflagellates, which are a group of living algae, eukaryotic algae, the nucleus does not break down. And the microtubules don't radiate out from the poles. Instead, they're parallel and they run right through the nucleus. And the nucleus itself elongates. So microtubules are made of protein. They're hollow tubes and they act kind of like railway tracks. And everything can be moved along them, the chromosomes, and in this case, the actual nuclear envelope itself. So in dinoflagellates, the nucleus inside stretches out, the chromosomes attach to the microtubules, and eventually the nucleus itself will pinch off into two separate nuclei, and then the cell will pinch off into two separate nuclei. There are other eukaryotic algae known as diatoms where the spindle has moved inside of the nucleus. It's found at either pole of the nucleus and it radiates out from those poles. Interestingly, it looks just like the mitosis you might be familiar with, but it's happening inside the nucleus itself, and the nucleus is going to stretch and pinch off. And in most eukaryotes, what you're seeing down the bottom there, what we've just talked about on the right, the nucleus does break down, and that happens during prophase. This is interesting where we see all these stages in living cells today, and we can work out from what we see today how the different steps might have evolved. It's very, very similar to what we saw with the eye. If you remember back to when we talked about the eye and the evolution of the eye, we mentioned that there are several different forms of the eye that exist today, and they tell us how this transition, the development of the eye, might have occurred. And here we're seeing transitional forms that tell us how the evolution of mitosis might have occurred. Mitosis in eukaryotes has a few different functions. Again, this is acellular reproduction. At the top left there, you're seeing two amoeba. Not that long ago, we had one amoeba, and that amoeba has divided into two through mitosis. So this is a form of reproduction, making copies of yourself, making clones of yourself. However, amoeba will occasionally have sex as well. And as mentioned, we do see this a lot. We see many species where asexual reproduction is the norm because it's quick and easy. You don't have to find a mate, but occasionally they will have sex so that they can mix their genetic material with someone else's, and we'll talk about why that's important. So in a lot of, especially single-celled organisms, mitosis is the main form of reproduction. On the top right, we're seeing a blood cell that is dividing. And blood cells are produced within the red bone marrow of your body, so in your long bones, things like your humerus and femur and so on. And here we're seeing the replacement of worn out cells. So as your red blood cells wear out, they need to be replaced, and that's done by the mitosis of healthy cells. And down the bottom there, we're seeing development, specifically in a sea urchin. So a sperm and an egg cell have come together, and they've created one diploid cell, and then that cell has undergone its first division. It's amazing to think that you came from one cell. So the fusion of the sperm and the egg creates a cell called the zygote. That's a diploid cell. And then that cell undergoes division after division after division, divides into 2 cells, 4 cells, 8 cells, 16 cells, etc. until it becomes the 30 or so trillion cells that make up you. As we'll see as we go through this course, where mitosis and meiosis occurs within the life cycle of an organism helps us place it within major taxonomic groups. Mitosis always conserves the ploidy number. If we start out with a diploid cell, then that cell will divide and split into two cells, and they will both be diploid. That's what we see for most of the cells in our body. But I should point out that the first eukaryotes were haploid. Bacteria are haploid, in fact. They only have one set of information. But the very first eukaryotic cells had one set of chromosomes, and they would divide just by mitosis. Our diploid cells in our body divide by mitosis. Skin cells replace worn-out skin cells. Blood cells replace worn-out blood cells by mitosis. However, in plants, there are weird cases where we have triploid cells, they're 3N, or we have pentaploid cells, they're 5N, and those cells can reproduce by mitosis and give rise to cells with the same ploidy number. Meiosis, on the other hand, always cuts the ploidy number in half. So if we start with a diploid cell, we end up with 4 haploid cells. In some plants, the cells are tetraploid. That means they're 4N. If they undergo meiosis, we would end up with 4 haploid cells. So again, the take-home message here, mitosis keeps the ploidy number the same. The daughter cells have the same ploidy as the parent. The daughter cells have the same exact information as the parent. They're clones of the parent. And meiosis always cuts the ploidy number in half. And also meiosis, because you're taking DNA from 2 different individuals, those individuals have the same genes, but they have different alleles of those genes. With meiosis, you end up with cells that are half the ploidy and are also different than the 2 starting parents. They're a combination of the contributions from those 2 parents. I mentioned before that chromosomes are made up of a mixture of protein and DNA, about half protein, half DNA. The DNA contains the genetic code. It contains genes. Remember, each gene codes for a different protein. But the DNA is wrapped around proteins called histones. And what the histones do is they protect the DNA. It is a rather fragile molecule, and they allow it to be very, very condensed during replication. So the DNA molecule is not broken when we're moving chromosomes from the middle of the cell to one of the ends of the cell. The DNA is a fragile molecule, so it does need this support. It needs this scaffolding from the proteins. When DNA is fully condensed in a chromosome, it's shrunken down to about 1/10,000th of its original length. And in fact, this is a pretty incredible statistic here. But if you were to take the DNA that's found in one nucleus, in one cell in your body, let's say a skin cell, and you took all the bits of the DNA and you put it end to end, you would have about 3 meters of DNA in one cell. So the proteins have to condense the DNA just so it will fit in the nuclei of your cells. By prophase, the DNA is completely condensed as much as it can be. And what we're seeing here on the right is a chromosome that consists of 2 pieces. And I just want to make sure you understand that what we're seeing is a mixture of DNA and protein. we would have one strand of DNA here. And I'll try to draw this somehow. But we would have a strand of DNA and it would be all wound up in here, a lot more wound up than I'm able to show with my scribble. But we have one big, straight, long piece of DNA here connected to proteins that help protect it. Remember, before division began, that DNA had been copied. So on the other side, we have a copy of that DNA. And again, it is all wound up inside this chromosome. So this particular chromosome has two pieces of DNA. And if everything's gone right at this point, those two pieces of DNA should be identical. One thing that can be really confusing when it comes to learning this stuff for the first time is the terminology. Chromosomes are made up of DNA and protein. The actual material is referred to as chromatin, but chromosomes can consist of one piece of DNA or two pieces of DNA. So again, we have a chromosome up here that consists of one piece of DNA that's all wound up and twisted up, and it's wrapped around these histone proteins, and the proteins stick together. And down here, we have a chromosome that consists of two pieces. We still call it a chromosome, though. So we have our original piece of DNA, and it's being used as a template to make another copy. That occurs during S phase of interphase. And then we end up with our chromosome here that consists of two chromatids. So at the top, we have a chromosome that's just one piece. In the middle there, in that black box, we have a chromosome that consists of two sister chromatids. They're called sister chromatids because they contain the same sequence of DNA. And then down the bottom here, after these chromatids have been pulled apart, once again, we have chromosomes that consist of just one piece and not two. Mitosis is asexual reproduction. If meiosis is involved, it's used generally to produce gametes that can come together to produce a new individual, and that is sexual reproduction. Sexual reproduction allows for the combination of alleles and the traits they code for from two individuals. Basically, by putting two sets of information into one individual, we have more diversity of information to draw from. Sex appeared in early eukaryotes over 2 billion years ago, and it allowed for the combination of alleles and the traits they code for from two individuals. So if you have two sets of information that are slightly different to draw upon, you have a greater diversity of genetic material and possibilities. Apparently, sex is very helpful for survival because, of course, evolution and natural selection have favored eukaryotes that engage in sex. Meiosis has made sex possible for a very long time. There's a famous saying in biology that was presented by a man named Samuel Butler, not actually a biologist. He was an English scholar and a friend of Darwin's, and later kind of an enemy of Darwin's. But anyway, he said, a hen is only an egg's way of making another egg. Kind of philosophical there. Which came first? But what he is stating here is that it's the gametes that are the most important cells in eukaryotic evolution. The gametes contain the genetic material, and the adults that produce them are basically just vessels to create these gametes and bring them together. The gametes that are brought together create the zygote. They create the next generation. Here's an overview of the role that meiosis and sex plays in animals like ourselves. This is the generalized animal life cycle. Down the bottom, we have a diploid multicellular organism, a mouse in this case, but it could be a human. And what that means is the cells have two sets of chromosomes, and this organism is made up of many of those cells, trillions of those cells. These diploid cells are not all capable of undergoing meiosis. Only a small number of cells within the gonads, within the testes or the ovaries, are capable of meiosis. And those cells will undergo meiosis and produce haploid gametes, sperm or egg. Now, if sperm or egg come together, that's something known as fertilization, also known as syngamy. Syn means to fuse, so we're talking about S-Y-N here. Think of synthesis, for instance, gametes. So syngamy or fertilization, that's where the sperm and the egg come together, and that produces a zygote, which is a single diploid, so 2N cell. And then that cell in the mother, in the uterus, for an animal like ourselves, will undergo many, many rounds of mitosis to give rise to a baby that, of course, is born and becomes an adult. So we spend most of our lives in a multicellular diploid state, and we have a short-lived haploid unicellular state, and that's just the gametes. We'll go through each of the stages of meiosis briefly, but before we do, let's compare meiosis to mitosis, and I'm going to use the same example that I used before. So in the middle here, I have a parent cell, and this cell has the condition 2n equals 4. What that means is the cell is diploid, has a set of information from two parents, so two sets of information, and the total number of chromosomes is 4. As you can see, you can count them. If we were talking about a human diploid cell, like a skin cell, a muscle cell, whatever, the condition would be written out 2n equals 46, because in our diploid cells, we have two sets of 23 chromosomes. Okay, so those chromosomes right now each consist of a single piece of DNA, but they're going to replicate. And in prophase, you can see that we have chromosomes that have two chromatids. They're still the same number of chromosomes. We still have 4 chromosomes, but each one has 2 pieces of DNA in it. The chromosomes are going to align themselves in the middle of the cell, and then what happens next is the chromatids are going to be pulled apart to opposite ends of the cell. And we end up at the end here with daughter cells that are identical to the parent cell we started with. So we have 2 diploid cells. So in mitosis, we have conservation of the ploidy. The ploidy doesn't change. We also only have 1 division, and we end up with 2 cells in the end. Let's take a look at meiosis now. So we're starting with our same parent cell. We have chromosome replication again, this time towards the right, and you can see something weird is going to happen. So just like we saw in mitosis, we're going to end up with 4 chromosomes, each one consisting of 2 chromatids. But the homologous chromosomes, the ones that are the same length, have the same genes, they're all going to come together to form what's called a tetrad. Each tetrad has 4 chromatids. And now bits and pieces of DNA are swapped between those homologous chromatids. And we end up with this exchange of material. You can see that happening during metaphase 1. And then what happens in the first division is the chromosomes are separated. And in the second division, the chromatids are separated. We end up down the bottom here with daughter cells that are haploid. So the ploidy has been cut in half. That always happens in meiosis, and it takes 2 divisions to do that. But keep in mind that the DNA is not copied between those divisions. Also, of course, the cells we end up with are not the same as the cells we started with. They have half the ploidy, of course, but also the chromosomes that were in the original parental cell have had their bits and pieces kind of reshuffled together. We haven't created any new information, but we've put bits of information, alleles, that were on different chromosomes together on the same chromosome. Okay, so the stages of meiosis, just very briefly. So right after the S phase of interphase, we have something called the G2 phase. And all I want to point out here is that the DNA has been duplicated. If you looked at the cell under a microscope, though, the nuclear envelope is still there. The DNA is very, very loose. You wouldn't be able to make out individual chromosomes. But the DNA has been duplicated, and those DNA strands are sticking close together. Because there's 2 divisions in meiosis, we refer to it as meiosis 1 and meiosis 2. So in the first meiotic division, we have prophase 1, and during prophase 1, the nuclear envelope breaks down, and also the chromosomes, homologous chromosomes, come together to form those tetrads. And when the tetrads form, we get these points of crossing over. They're referred to as chiasma or chiasmata. Don't worry about that, but these are just points where bits and pieces of information are being exchanged. By metaphase of the first division, so metaphase 1, the chromosomes are still attached to each other in tetrads, and they've moved to the middle of the cell. The chromosomes are still kind of tangled up, so they have swapped bits of information, but the DNA still has to be broken and then resealed in order to get the chromosomes apart. But they've attached to the spindle and they're ready to move. The chromosomes release from each other, and we can see in anaphase they're moving towards either pole. And note that we've had bits and pieces swapped between homologous non-sister chromatids. During telophase 1 and cytokinesis of the first division, the cell is elongating, it's pinching off into 2, and the nuclear envelope is reforming. Now that was the first division of meiosis. Now we have a second division. And the second division is a lot like mitosis, really. So what happens now is the nuclear envelope is going to break down again. The chromosomes, which consist of 2 chromatids, are going to move to the metaphase plate in both of the cells, and then, like we see in mitosis, they're going to be pulled apart. In anaphase 2, the chromatids are being pulled apart, and once they've separated, we refer to them as just chromosomes again. And then during telophase 2 and cytokinesis, we get a nuclear envelope forming again, and we end up with our haploid daughter cells. This is our end product. The cells are genetically distinct because of that crossing over, because of bits and pieces of homologous genes have been swapped. And we're swapping alleles, we're swapping different forms of genes. We haven't lost genes or gained genes, but we've changed the alleles that are on the same chromosome. Back to the big question. Why bother with sex? Why does sex exist? Meiosis is more complicated, it's messier, there's more things that can go wrong. Why bother? It's not about reproduction. If I told you it was about reproduction, I'd be lying. Terrible, terrible pun on the photo here, which I'm not going to discuss any further. If your goal is just to make as many babies as possible, sexual reproduction is maybe not the way to go. Sex is messy, as we've talked about. It's complicated. Meiosis is complicated. There's a lot that can go wrong, and we need to have two parents that come together to produce those offspring. Asexual reproduction doesn't involve finding a mate. It doesn't involve meiosis. You're just making a clone of yourself. Now, it might surprise you to learn that there are many vertebrates, those are organisms with a backbone, like us, for instance, that can actually reproduce asexually. One example is lizards. There are some species of lizards that can reproduce sexually or asexually. They have a choice. If they're reproducing asexually, they just make a clone of themselves, and the entire population is female. Now, how does that work? We've talked about the formation of a zygote before. They still form a zygote, sort of, but what they do is they have cells in the ovary that undergo mitosis to produce a diploid cell, and then that diploid cell develops into a new lizard. With sex, of course, if these lizards decide to have sex, there has to be males and females. The males produce sperm, the females produce egg. That comes together to produce a diploid zygote. But regardless of whether it's sexual or asexual, the zygote is going to develop the same. So let's compare the two strategies that we see in these lizards. So with sexual reproduction, we have to have two individuals coming together to produce offspring. With asexual, every single lizard is female, and they produce their zygotes by mitosis, and they produce clones of themselves. Now let's just pretend, just for the sake of argument, that every mating opportunity, every reproductive opportunity results in two offspring. If we look at sexual reproduction, we would have two individuals coming together and producing two offspring. The population would not grow in size over time. We would just be maintaining the population, best case scenario, but due to predation and due to just death in general and bad luck, the population would probably shrink in size. Now, in the asexual situation, every single lizard can produce two copies. The population would grow over size. So again, it's kind of an artificial, hypothetical example I'm giving here, but you can see that with asexual reproduction, every single individual can produce potentially quite a few offspring, whereas with sexual, we have to have two individuals coming together to produce anything at all. So again, with our example here, where every reproductive event results in two individuals, our asexual population would grow very quickly. Our sexual population would not grow at all. In fact, like I said, it might shrink slightly. So why on earth would we want to have sex if the point is to make copies of ourselves? If we're looking at evolution, we're looking at species competing with each other and trying to dominate over other species. Why would we favor sexual reproduction over asexual reproduction. The reason is kind of complicated, but the main reason is variability. If you are reproducing asexually, you are making a clone of yourself. Every individual that you spit out is going to be exactly like you. Now, if you take your information and mix it with someone else's information, you're going to have offspring, babies, children, that are different from either of you. In fact, every single individual that you produce is going to be a little bit different. Maybe you're really successful in a Darwinian sense, in a fitness sense, but maybe you're not. If you can spit out children that are all slightly different and the environment changes a bit, some of them are going to have advantages that you didn't have, that your mate didn't have. It's going to lead to a lot of opportunity. So what we're seeing here, these are aphids. These are little tiny insects that you might have come across if you've looked very closely at plants. They're very common. And what they do is they live on plants. They have little piercing mouth parts, kind of like little hypodermic needles, and they'll stick that into the plant and they'll drink the plant's juices. I mean, if you're a gardener, if you're growing roses and stuff, you probably hate these guys, but they're everywhere and they feed a lot of stuff. I think they're pretty cool. They are fed on, they're eaten by a whole bunch of different things, and they reproduce as quickly as they can to survive. What you're seeing in the bottom left is a female aphid that is giving live birth to a new aphid. During the summer months when things are good, all of the individuals are females. We have females that give birth to new females. They do that asexually. They produce zygotes by mitosis, and they spit out this new baby that's a clone of them. But what's really interesting is at the end of each season, late fall, they will produce some males. The females will produce a few males, and the males will mate with the females. And when they mate, they produce eggs. And that's what you're seeing in the lower left. Those eggs will survive the winter, and they'll give rise to the next population next winter. So during the summer, things are really, really good. The whole strategy, the goal of the aphid is to produce as many babies as possible. They do that asexually by giving live birth to new females. It's a process known as parthenogenesis. It's really, really fast, so they can spit out as many new babies as possible. In the fall, they engage in sex, which is a slower, messier, more complex process. They have to go through meiosis to produce sperm and egg. The females and males have to find each other, come together. But they produce these eggs that last through the year. The eggs, again, are the result of sex. So sex is where we have two partners coming together to produce new individuals. But next spring, when these eggs hatch out, every single individual is going to be a little bit different. And that's key here, because who knows what it's going to be like next year. Maybe next year certain plants are going to be readily available while others aren't. Maybe there will be more predators of one type versus another. Maybe if you have a slightly different color, you might be better camouflaged because there's different types of plants, different predators that can see different things. Who knows what's going to happen next year? So having that variability means that you're not putting all your eggs in one basket, so to speak. Hopefully, some of your babies are going to succeed next year. This is a theme for a lot of invertebrates. If conditions are good, there's lots of food, they're happy, they're going to reproduce asexually because it's faster. They don't have to find a mate, they don't have to do meiosis. What you're seeing at the top here on the right is something called a hydra. And a hydra is like a sea anemone, but these things are small. They're about a centimeter in length, but they have the same kind of structure. So at the top of this critter, we have tentacles that can grab onto stuff. We have a mouth in the middle that will eat stuff. And these are things that you find in local ponds. If you go out in the summer and take a look at the underside of a lily pad or something, you'll see lots of these little tiny guys, if you look closely, attached to the underside of the leaf. They reproduce asexually, and that's what you're seeing here. So we have a little bud, a new individual that is literally just growing off of the body of this hydra. Imagine if you could do that. Imagine if you could grow a new human asexually off of your hip. It would grow and then it would just pop off and go about its life, do its own thing. That's what we're seeing right here. And that's how these hydra reproduce for most of the year. Now, once the lake gets cold or the pond gets cold and we're entering into winter, what they do is they produce males and females. The males and females mate. And just like we saw with the aphids, they produce these eggs that will withstand the harsh conditions of the winter and give rise to new individuals next spring. Again, the whole point is we want variability because we don't know what things are going to be like next year. Things are always changing. If we look at mammals like ourselves, we do this as well, but we do it all the time. We don't switch between sexual and asexual reproduction. We've just focused on the sexual reproduction. What you're seeing on the bottom here, this is a bit of an outdated a diagram here, but this is from one of the earlier versions of your textbook. It's been estimated that any pair of humans can produce about 8 billion or so different offspring, different children that are different in some way. That's larger than the population of humans on the planet. So if you pair up with a mate, you can have 8 billion or so different combinations of offspring. A huge amount of variability. They're different from you and they're different from your mate. They are a recombination of you and your mate. And that is due to the recombination that occurs during prophase 1 of meiosis. Now on the far bottom right, I've got a picture there of two spiders. They're actually of the same species. So a lot of spiders, the black widow spider would be an example of this. The female and the male are quite different. This is something known as sexual dimorphism. Di means to, morph means form. The sexes are different. The sexes are different in humans as well. It's not as pronounced as it is in a lot of insects where the female is very, very big. She's big because she needs to lay eggs. That takes a lot of energy to do that. Egg cells are very big. And the male is typically much smaller. And in something like a black widow spider, the male and female do things that are somewhat different. So the female and the male might have different life strategies. In the case of black widow spiders, as you've probably heard, the female will try to eat the male after it's mated, because after it's mated, you're not that much use to the female, really. It's already done its job. But the male, if it's really, really clever and really, really quick, it might be able to mate with a couple of females. So we have a situation here where the males and females have different traits that are being selected for, but they're both traits that are helpful. So we have a situation where having males and females performing slightly different roles helps the species or helps the offspring on the whole, because we've got different selective forces acting upon the male and the female that are selecting for different positive traits that are both passed on to the offspring of the two. There's one other thing to consider. So variability is the big thing here. variability and the fact that males and females may do different things and may evolve in slightly different ways that can help both males and females. But the other thing is that when you have sex, you have an opportunity, the only opportunity you have, to compare your genetic information to somebody else's information. And you actually have a chance to correct for any deficits, any problems. When you produce your gametes, basically what you're doing is you're taking information from your mother and your father. So let's say that on this diagram here, your mother's information is in red, your father's information is in blue. That information has been separated in your cells up to when you make your own gametes. In the cells that undergo meiosis, the information from your mother and from your father come together, and we have the swapping of bits and pieces. It's the only time this occurs in your body. This creates gametes that are different, and of course your mate is going to produce gametes through the same process. They come together, we get a zygote that's got a different arrangement of genetic material than either parent. But one thing that's kind of interesting that happens during your making of gametes is that you have this opportunity to compare your father's information to your mother's information. The genes line up right alongside each other one by one, and your body can look for errors. Maybe there's a gene that has a mistake in it, and that can be compared to another set of information, another gene from your other parent. And there's a chance here for your cells as they're making gametes to correct mistakes, to kind of proof the information, to check it against other information. So meiosis also allows you a chance to proofread, double check your information and make sure it's okay. And there's one last chance here to correct mistakes before that information is passed down to the next generation. And once again, that's what we're seeing here. We're seeing crossing over. We've got information from your mom, information from your dad, and this is being compared. And there's one last chance here for enzymes such as DNA polymerase to correct any mistakes that there might be. Once again, the main purpose of meiosis is to cut the ploidy in half. Imagine if we didn't do that. Imagine if we created diploid gametes, diploid sperm and egg. When they came together to create a zygote, the zygote would be 4N, or tetraploid. And let's say that that generation did the same thing. They created gametes that had the same ploidy. 4N, they'd come together, they'd produce a new generation that was octaploid, 8N. This would spiral out of control, and pretty soon all of our cells would be occupied by DNA. They'd be mostly DNA. So every generation we need to cut the ploidy of the gametes down to half the value of the cells that made them so that we can recreate the diploid condition in the next generation. This process of meiosis occurs at different points of the life cycle for different organisms. We'll look at animals first. We're animals, and this is actually the simplest process. This is the simplest life cycle. In humans and other animals, we have ovaries and testes that produce haploid gametes. They come together, and that produces our zygote. So that process where the sperm and the egg come together is known as fertilization or syngamy. we get a diploid zygote that's going to develop through mitosis into the adult. So the diploid zygote is going to divide to give rise to 2 cells. Those divide into 4 cells. Those divide into 8 cells, etc., until we have the 100 trillion or so cells that make up the adult. Now, this is a bit of a prelude to our next topic. It's thought that meiosis is just a really strange and really broken mitosis. It was a happy accident. The first eukaryotic cells that engaged in sex were almost definitely single-celled organisms. They were single-celled and haploid. So we have these haploid cells swimming around in the water, and occasionally they'd find each other and fuse, so 2 cells fusing to create a diploid cell, and then immediately undergo meiosis to give rise to 4 haploid cells. So 2 cells come together, diploid cell, immediate meiosis, back to haploid, but this time we have 4 cells instead of 2. So it was a way of reproducing, but also they got the benefit of checking their DNA against someone else's DNA, of having 2 sets of information combined in one individual to increase diversity and increase potential. In animals, the diploid stage became multicellular, and that became the dominant stage. In fungi, though, the exact opposite occurred. The haploid stage became the dominant stage. So if you see mold growing on a piece of bread, that is a multicellular individual. It's a multicellular fungus, but the cells are haploid. If you look at, let's say, a toadstool or a mushroom, you might realize that they have a cap-shaped structure, kind of like an umbrella, and under that cap, there are diploid cells, tiny little diploid cells in there, and they're produced and immediately undergo meiosis to take us back to the haploid multicellular stage. So the diploid stage was favored in animals, the haploid stage was favored in fungi, and plants are just really weird because they have a multicellular diploid stage and a multicellular haploid stage, as we'll see. And a few concepts here to end things off on. We are animals. Animals are multicellular and diploid. Diploid means that we have 2 sets of chromosomes. So once again, for us specifically, for humans, we have 2 sets of 23 chromosomes in all of our cells except for the gametes. Gametes are haploid, and when the haploid gametes come back together, they reform that diploid state. So we have the ploidy being cut in half and then reconstituted for every single generation. I put usually there because there's some plants and some other organisms that can do some weird things. We might touch on that a bit later, but easiest for now just to consider that gametes are haploid. Mitotic divisions, so divisions of mitosis, conserve the ploidy. It doesn't matter what the ploidy was that the cell started with, we're going to get the same ploidy in the daughter cells. Now in humans, we only see mitosis in diploid cells. So we're always going to start with a diploid cell and we're going to get a diploid cell out. Meiotic divisions, though, cut the ploidy number of the cell in half. So in humans and other animals, we start with a diploid cell, we end up with haploid cells. And finally, some terminology. I was rather tired when I recorded this, and I did record it over more than one sitting, so I hope I have not repeated myself. If I have, if I've been really redundant, my apologies. And I'll see you next time for the protista.